Hello everyone and welcome to the webinar on Rhagoletis pomonella of the Group 1 Fruit Flies EU Priority Pest. As you might know, we previously provided 3 webinars on the EFSA Plants Pest Survey Methodological Framework and the toolkit, including the Ripest and the OptiPest, and the 3 webinars of the Group 1 Fruit Flies Trephididae. If you did not attend the previous webinars and you would like to know more about them, the recorded presentations are available on the EFSA's YouTube channel and on the EU Academy website. This is the last webinar of a round of 4 that will take place before the summer break, and today's presentation will refer specifically to the pest Rhagoletis pomonella. Lastly, for the new attendees, and as a reminder, since the microphones are mute, the Q&A chat is enabled for you to ask any questions that might come up while the experts are presenting. Before we start, I would like to introduce some housekeeping rules. You are automatically connected to the audio broadcast, one-way audio, just listening mode, so you cannot speak. This event is in English, and questions should be submitted in English via the Q&A chat. If some of the questions remain unanswered while this webinar, you can resubmit them via email. This event is being recorded, and recordings will be published on the EFSA's official YouTube channel and in the EU Academy. We will send all the information after this webinar. After the event, attendees will be requested to evaluate the EFSA's event filling out Microsoft survey, so we can know if we can improve for the next webinars. As you might see in your screen right now, this is the agenda for today. Now we are in the introduction, and in a few minutes, I will give the floor to the 2 experts on Rhagoletis pomonella that we have today, Mark Kinkar and Joseph Jacques. And after their presentations, we will have the short quiz, as I told you, with Mentimeter, just to know if you understood all the presentation. And by 11 o'clock, we will get to the end of the webinar. So I'm Jose Ovejas, I'm a journalist, and I will be the moderator of this webinar. And with us today, we have Mark Kinkar from the EFSA's Pest Survey Methods Working Group. He is from the Estonian Agriculture and Food Board at the Department of Plant Health and Propagating Material in Saku, Estonia. And some of you already might know Joseph Jacques. He was in the previous webinar. He's a former professor of plant production at Universitat Jaume I. Now you should see in your screen the Mentimeter. You have there the QR code, as I said, so you can scan it with your mobile, or you can either go to the website and and you have to write down www.menti.com. We would like to invite you all to participate in this poll. So we recognize who is in the room. Please try to search www.menti.com or scan the QR with your mobile. To participate, you just have to go in there, and if you go, you'll see in the upper side of the slide a code that you can introduce, and you'll see the poll. It is advisable for you to keep this in mind because we will use Mentimeter again at the end of the presentation to see if you understood the webinar. Okay, so sorry for this. Let me give the floor to Joseph Jacques and Mark Kinkar. The floor is yours.
Okay, good morning, everyone. And good morning, Mark.
Good morning.
Okay, so basically, Mark, I need your help because I have to design a survey for a pest. So how should we proceed? Do you want me to start with presenting you some data on the biology of this pest, for example, and then maybe you could talk about the application of the framework for Rhagoletis pomonella?
Absolutely, sure. Let's do that. So first, go ahead and let's get to know this fruit fly.
Okay, if we go to the next slide, then I'll introduce you to the pest. This is the pest, yet another fruit fly, the apple maggot. However, as I mentioned, this has quite unique features that differentiate it from other fruit flies, which are priority pests for the EU. Most of the information that I have used to introduce this pest comes from EFSA documents, including the pest survey card on Rhagoletis pomonella, which is accessible from the Ripest app, the story map on Rhagoletis pomonella, and the Rhagoletis pomonella pest report supporting the ranking of EU candidate priority pests, but also from the datasheet on this fruit fly from EPO. Here we can see a scheme of the life cycle of Rhagoletis pomonella and some important biological features of this fruit fly. As all holometabolous insects, Rhagoletis pomonella has 4 different development stages: the egg, the larvae, the pupa, and the adult. Adults emerge during late spring, around June, and after about 10 days feeding, they become mature and mate. Then, females can lay up to 400 eggs under the skin of apples during their lifetime, which is of about 1.5 months. They complete development in about 2 to 5 weeks, and then the mature 13-cell larva abandon the fruit and bury in the soil to pupate. Pupae most often enter a diapause that will continue until the following spring, when the following generation of this fly will emerge. So this is a univoltine species, only 1 generation per year. Pupation can also take place in the soil if protected by litter or debris. And although most of the pupa will remain inactive until the following spring, some may not stop and produce a partial new generation within the same season. And some may also extend their diapause for more than 1 year. However, this is not the rule. The rule is univoltine, 1 single generation per year. Importantly, this species requires frequent remating of the females for eggs to hatch correctly. And as you can see on the table on the right, females that don't remate frequently may result in egg hatching as low as 45%. So there's a high impact on the population of this species.
But Joseph, may I ask, how common is that some flies stay in the soil for 1 or 2 years and some emerge before the winter in the same year? And what's the cause of that?
The most common life cycle is the univoltine. Alternatives, like a second generation within the same year, most probably represents a suicidal generation. The hemivoltine life cycle has fitness costs that probably have led to the selection of the univoltine populations as the most fit.
Okay, thank you, Joseph, for that answer. I hope you can hear me. If I can ask one more question.
Yes.
So how would these features affect detection surveys of Rhagoletis pomonella compared to the other tephritids that we have seen in the previous webinars?
Okay, I've prepared a slide for that. And if we move to the next slide, I'll tell you. When we compare this fruit fly to other fruit flies like Anastrefa ludens or Bactrocera zonata, we see that there's differences in voltinism. Rhagoletis pomonella is mostly univoltine, whereas the other species are multivoltine. Rhagoletis pomonella has a diapause. Importantly, when they are in the soil, there's an obligate diapause, so they have to stay there until the following spring, minimum. Only some exceptions, but this is not the rule. In the case of the other fruit flies, there's no diapause. The life cycle continues as long as temperatures are fine. And also the cold tolerance. Rhagoletis pomonella is a little bit more cold tolerant than the other species. And this affects, on the one hand, the time of sampling, but in this case, there's no differences because we will sample for these flies at the same time in summer. But there's other effects which are more important. Because we cannot rear the larvae that we may collect in the field for Rhagoletis pomonella because they have to undergo this diapause. So if you, for example, sample fruits and take them to the lab, you cannot put them in the lab and wait for 1 week, 2 weeks to have the adults emerging. And this has consequences because there's no identification methods or suitable identification methods for the larvae. Additionally, because there's this diapause, and just 1 generation per year, we cannot rear this Rhagoletis pomonella as we do with the other species. So then we cannot apply the sterile insect technique for pomonella, which was an option for the other species. And this will also have effects when we detect it. And last but not least, there's a better adaptation of pomonella to colder climates, not because of the higher tolerance to cold, but because of this diapause that allows this species to continue and to endure, for example, the European winter in most of the places.
So how about the climatic suitability for this pest?
Well, if we go to the, later we'll see the climatic potential for this pest, but the important feature is that this is a univoltine species and has only one generation per year, and this is what will happen wherever it can survive, no more generations than one. And as I said, although pomonella is more cold tolerant than other tephritids, it is the winter diapause that gives it the possibility to endure cold temperatures. And for example, in the previous webinar, we saw that Bactrocera sonata couldn't continue or couldn't establish in places where temperatures dropped below 15 degrees. But this is not a limit for pomonella because thanks to the diapause, it can survive. And now, if we move to the next slide and look at the host range of these species, we'll see another set of differences. So this is an oligophagous fly that feeds on Rosacea, especially on the hawthorn, Grataebus, and apples. Indeed, hawthorn is the ancestral host for this Eastern North America species, and it just moved to apples when Europeans introduced this crop during the 19th century in North America. Since then, two separate strains are recognized, with the apple strain becoming invasive in other parts of its current range, also in North America. And because apples is the most important host in the EU, this is the host selected for detection surveys according to EFSA.
But in economic terms, what makes this pest so important?
Well, if you look at the images we have on the right, as we can see in the images, this insect damages the fruit with the oviposition scars that deform the fruit and may result in complete yield loss when larvae consume the fruit pulp. This fruit may fall prematurely. The management of this pest increases the production costs and, importantly, can result in reduced market access due to quarantine restrictions. And last but not least, this pest poses a challenge to its detection, as we will see later, and this may enormously increase the associated costs. We can see these differences compared to other fruit flies. As contrary to other fruit flies, Rhagoletis pomonella is oligophagous and feeds mostly on apples. Detection can focus on apples only. Moreover, in case of detection, efforts to eradicate could exploit sanitation more easily than with other foliferous fruit flies, and this makes sanitation especially useful against this fruit fly.
And what about the current distribution of the pest?
Okay, if we move to the next slide, we can see the current distribution of pomonella. And as I said, this is a pest originally from eastern USA, where it used to breed on hawthorn, but which moved to apples about 200 years ago. Since then, it has established on apples in western USA also, and some areas of Mexico and Canada. Interestingly, in those areas, which are newly colonized, genetic diversity is lower than in the central areas in eastern US.
Okay, interesting. Is apple the preferred host also for the hawthorn strain? What do you think?
No, it's not. But we should keep in mind that the apple strain derives from the hawthorn strain. And this is an ongoing process of speciation, so it could happen again. Why not?
So it seems like climate would not be a limiting factor for this pest, and the establishment in the EU could really happen.
Yeah. Well, according to the climatic suitability modeling, this could happen. And if we move to the next slide, I'll show you. Now, okay. Here we can see the potential distribution of Rhagoletis pomonella according to Climex, which is a model based on climatic variables of the places where the pest is known to occur. We can see that most of the EU, with the exceptions of Finland and most of Sweden, is suitable for establishment provided that hosts, mostly apples, are present. Because apples are widespread within the EU, establishment shouldn't be a problem in case that Rhagoletis pomonella enters the EU. This is also a huge difference with other fruit flies, which in case of entry could most likely establish in the south of southern EU member states only. So Mart, do you think that now you have enough data to design the survey for this pest?
You gave a nice overview of the pest and its biology, and we can start, I think, creating or designing a survey for this pest, creating an example for today. But in order to do that, we still need a few parameters to set for this pest. And I will also quickly go over a few key aspects of the EFSA statistical survey methodology. So first of all, we need to think about the type of the survey that we want to perform. So there are these three different kinds of pest freedom surveys: detection, delimiting, and buffer zone. So what all of these types of surveys have in common is that they can be used to find any evidence of pest freedom in your survey area. And that's also why the EFSA methodology and also the repest application can be used for all of them. So in case of a detection survey, the pest has not been previously found in the survey area, and the aim is to confirm the pest status and find proof of pest freedom. The delimiting survey is performed if you already have located an infestation and you want to know exactly the size of the infested area where the pest is circulating. And the aim of the buffer zone survey is to find evidence whether the pest has spread outside of the infested zone after you have demarcated the outbreak area. And as you mentioned, Joseph, the Rhagoletis pomonella has not been found outside of North America, so we will also focus on detection survey and substantiation of pest freedom in our example today. But why do we need a methodological framework in the first place? It's because it's really impossible to say with absolute certainty that a pest does not occur in your survey area. Otherwise, we would need to sample all parts of all the host plants and test them with a perfect diagnostic method, and that is just impossible. So what we can do is survey a part of the host population and then take that conclusion on the rest of the population. So we can really look at the target population as a big iceberg where we can only survey the tip of it. And by using statistics, we can also say how confident we can be that the whole survey area really is pest-free. And the parameters we need for designing these statistical surveys are the number of inspection units in our survey area, the target population size, sampling effectiveness, diagnostic sensitivity, the relative risk of the risk level, and also design prevalence. But I will go over these one by one. So in order to design a statistical and risk-based survey, you should be able to characterize your survey area in these, at least in these four different levels. So the first level is the survey area, is usually the whole of the member states if you're doing an official detection survey. Next is the epidemiological unit, which is the subset of the survey area and is defined as a homogeneous area where the interactions between the pest, the host plant, and other environmental factors would result in the same epidemiology, meaning that that all the hosts within the same epidemiological unit would have a similar probability of being infected by the pest. So Joseph, what do you think? Which epidemiological unit should we consider for Rhagoletis pomonella?
I think an apple orchard. Would it be fine?
Yes, I would agree because apple orchards would be really suitable. These are quite uniform and well-defined areas with susceptible and economically important hosts. And also data about the total size of the apple orchards should also be available in the survey areas or in other member states. So in our example, we will consider one epidemiological unit, which is all of the apple orchards in our survey area. Next, the inspection unit is the elementary unit on which the detection method is applied in the field. And they define also the size of the subdivisions of the target population, meaning that the total number of the inspection units is really the size of your target population. And these inspection units could be, for example, an individual tree or a group of trees, an individual fruit. In case of trapping is used, it could be an area covered by the trap or even the pest directly. And according to the size or measure of the inspection unit, also the sampling effectiveness and diagnostic sensitivity should be somehow quantified. So sampling effectiveness is the probability to capture the pest during an inspection if the pest is present in that inspection unit. And diagnostic sensitivity is the probability to detect the pest in the sample given that the laboratory sample is truly positive. And then method sensitivity is the multiplication of these two values and is the probability to correctly capture and identify the pest in the inspection when the pest is present. So Joseph, can you help me again? So which would be the detection and diagnostic methods for this fruit fly?
Okay, if we move to the next slide, I'll show you that we have two main detection methods. On the one hand, we have the sampling of fruit in a tree, so that the inspection unit would be an individual tree. Symptomatic fruit, including those recently fallen to the ground, should be sampled, and this is quite time-consuming. The sampling effectiveness of this method was estimated at 30%. However, this method has one important drawback, and it is related to the identification of the pest. So far, morphological identification of the larvae is not possible, and the rearing of the collected larvae to adulthood is also difficult because of the univoltinism and the obligate diapause of this species. Because molecular methods are not conclusive for this species, the positive identification of Rhagoletis pomonella as a causing agent of the symptoms is not possible. Even though the association between apples and Rhagoletis pomonella is a strong evidence, the symptoms could have been caused by other fruit flies, like Keratitis capitata, which is present in the EU. Therefore, this method cannot be used for detection as a standalone detection procedure, but could be combined with other detection methods. And indeed, EFSA is currently working on this type of sequential sampling, and the app may be able to deal with these situations in the near future. So stay tuned and check EFSA website for updates. The other detection method is a trap, most often a yellow sticky trap baited with a female attractant, ammonium salts. The main problem with this trap is its extremely short coverage, which is about 2.5 meters, which makes that the area covered by a trap is roughly one tree. In this case, the sampling effectiveness was also estimated at 30%, and in this case, the morphological identification of adults is close to 100% efficacy. That's why, at this stage, we should focus on this type of traps for the detection of Rhagoletis pomonella.
Okay, but this situation is quite different from other fruit flies, right?
Yes, and if we move to the next slide, I'll show you again. We have the table where we compare pomonella and other fruit flies, and there are important differences. So you can see that on the one hand, the diversity of attractants for other fruit flies includes sex pheromones and allows the attraction of both males and females from a longer distance. On the other hand, the difficulties on the identification of immature stages can be sorted out by rearing those larvae to adults for the other fruit flies, so that morphological identification of the adults is possible even when sampling fruit. So these differences affect the availability of some control measures, like the sterile insect technique or the male annihilation technique, which can be applied to other fruit flies, but not to pomonella. And this will affect, in case of detection, what we can do.
Okay, thank you. Now moving on, let's talk about the risk factors also. So the level 3 of the population structure. So a risk factor is any factor that increases the probability of infection by the pest in a certain area. And focusing more on these high-risk areas allows us to reduce the total sample size needed to achieve the same design prevalence or the level of pest freedom. And this will hopefully also see in our example today. So in order to know where these areas of higher risk are located in our survey area, we should first know what the risk activities are that increases the chance of the pest occurring. And then identify the risk locations where these risk activities occur. And risk areas are the areas around the risk location where the pest may be found. And these areas are usually calculated taking into account the spread capacity of the pest. And in order to use the risk factor, we also need to know the different risk levels associated with the risk factor. For example, high risk, medium risk, baseline. We need to know the proportion of the target population to which the risk levels apply, and also the relative risk of each risk level compared to the baseline risk. So, Joseph, which risk factors could we use for Rhagoletis pomonella?
Okay, if we move to the next slide, I'll show you. So Rhagoletis pomonella has a lower spread capacity relative to other fruit flies. And this feature affects the size of high-risk areas, as we'll see, but also the size of the maximum eradication area that we will also see later. So we'll move to the next slide. In the case of Rhagoletis pomonella, high-risk areas, in this case, correspond to entry points, packing and sorting stations where apples imported from regions where the pest is present are processed. These areas are the ones that could receive infested fruit. In this case, infested fruit should remain undetected, and larvae within this fruit should be able to complete development, pop out the fruit, find a suitable place to pupate, endure the diapause, and then the following year should be able to emerge, find a mate, and then a possible host to oviposit. All this should occur within a 600 meters radius, which is the maximum distance this fly can cover in 1 year. So this is not that easy. The ECA recently performed by EFSA concluded that the risk within that area would be 2.35 times higher than in the rest of the area. So this is the relative risk of these areas close to entry points.
Okay, thank you, Joseph. I think that now we almost have all the data needed to design a survey for this pest. We have characterized the survey area, and the survey area for our example will be a made-up country named Surveyland. We will use one epidemiological unit, which is all of the apple orchards in our survey area. We have identified one risk factor, so we consider having higher risk of introduction near the possible entry points, packing and sorting stations of host fruit imported from regions where the pest is present. And we use trapping as sampling method, which also defines our inspection unit, which is the area covered by the trap. So, but due to the low efficiency of the traps, unfortunately, the size of the inspection unit is very small. As you said, only about 20 square meters. So we estimate that in our survey area, there would be about 20,000 hectares of apple orchards. That equals to 10 million inspection units. So sampling effectiveness, as you said, 30% for trapping, diagnostic sensitivity close to 100%, relative risk, we consider that 2.35 in areas close to the risk locations, in 1% proportion of the whole target population. So these are the main parameters we'll be using for our example, but there is still one parameter we need to set, and that's the design prevalence. So which is the threshold of the pest prevalence that the survey would be able to detect. So the design prevalence, also defined in ISBN 31 as evidence of freedom, is the maximum prevalence of the pest that could go undetected within the survey design. And of course, lower levels of design prevalence will result in higher numbers of sample size if you still want to achieve the same level of confidence. And this is also depicted on the graph on the right here. So setting the design prevalence level as well as the desired confidence level is usually a compromise between the level of risk that you're willing to take and available resources. And one way to think about it is also what is the maximum prevalence the pest could have reached and still be eradicable. So, Joseph, do you have an estimate also for a possible estimate for design prevalence?
Yes, we could base it on the maximum area of Rhagoletis pomonella infestation that could still be eradicated, which was recently estimated by EFSA at 1 kilometer radius, which means 314 hectares. So do you think that we could use this value to define the design prevalence?
Yes, I think we could. But may I ask, why don't you include, in this case, common control methods against fruit flies like the SIT or the MAT?
Yeah. If we move to the next slide, I'll show you again the table and the comparison, and we'll see that in this case, Rhagoletis pomonella have different features that preclude the application of some methods which are commonly used against other fruit flies. On the one hand, we have the voltinism that prevents the application of SIT, the sterile insect technique, in pomonella, whereas it can be applied to other fruit flies. The host range makes that sanitation especially suited for pomonella, but more difficult to apply to other fruit flies which are polyphagous. Then we have the availability of attractants, which is wider for other fruit flies, but in the case of pomonella, we only have a female attractant and with a very short coverage, which means that we cannot apply mass trapping and we cannot apply MIT, the male annihilation technique, in pomonella. And finally, because this is a wake flyer, just 600 meters per year, compared to other fruit flies, this compensates a bit, and the size of the maximum eradication areas is still 1 kilometer, which is the same as for other fruit flies. I hope that now we have been able to follow all the explanation.
We have. Thank you. Thank you very much.
Thank you.
So we can use this 1 kilometer radius, and that would mean that our design prevalence, so the area of pest presence that would still be eradicable, would, in our example, would be 1.6. So that's 1.6% of the target population that will be able to take the risk. And now I think we have all the data we need to create this example in repest application. So I will just open my web browser here. But before I open the repest application, I just wanted to show you something very interesting, and that is the pest surveillance database. And EFSA has quite recently updated a new feature in the relational database, and this is this pest-focused visualization. So if I select the pest of interest, for us is Rhagoletis pomonella, I can see a nice overview of all the different parameters that have been entered in this relational database. So this this is all the available data that is available for this pest from the pest survey cards and different expert knowledge elicitations. So as you can see, I can select the host that I'm interested in, and I get information about the sampling months, the spread type, spread capacity, about risk factors, relative risk of the different risk levels, also the sampling effectiveness for different sampling methods, and so on. So it's a very useful place to get all the information available from this one place. So you have this overview table in here, and you can also go do a deep dive search in here of the things that you're interested in. So I can really suggest people who are interested to have a look at this new feature in the surveillance database. But let's have a look now in Repest. We will try to design a survey as we have explored today within this presentation. I have already uploaded all the data in the Repest tool, so I can show you these different calculations and see where we end up. So right now I'm in the initiation phase. So the motivation of the survey is to comply with the regulatory requirements. The target pest is Rhagoletis pomonella. Let's say that the status of the pest is absent, and last year a detection survey was also carried out in our survey area. So here you can select the survey area. As I said, where the country for us is Surveyland. It's a made-up country in the Repest, just for these kind of experiment occasions. So I have selected all the 6 NUTS1 regions in our Surveyland, and we can move on. So which host plants are targeted in our survey? We have apple here, and we don't have a shapefile for the host plant distribution. So we're entering the preparation phase. We can skip these parts. The inspection unit for our case is the area that the trap covers. So we have selected area from the drop-down menu. As for the epidemiological units, I have inserted one user-defined epidemiological unit, which is all of the orchards in our survey area, and the number of inspection units is 10 million. It's very important not to forget to insert the number of inspection units here. So we have one risk factor. We have labeled it accordingly. You can see the database options are already here that I can quite conveniently choose for this pest. There are no other risk factors. Type of the survey site is orchard. Survey period we have entered here. How many detection methods will we use? We're using trapping, so one detection method. Sampling effectiveness 30%. Diagnostic sensitivity, we're using 99, which is close to 100%. And we can continue. So, and now we're in the most important page. The overall confidence that we want to achieve is 95, which is the sort of default value for confidence level. Design prevalence is 1.6. We have 2 risk levels within our 1 risk factor. We have high risk area with a relative risk of 2.35 and 1% of the whole survey area. And we have our baseline area. So if we click on simulate, we can see that the theoretical sample size would be 136 from the high risk area and 319 from the baseline area. And that would give us the same kind of confidence in both of the risk levels. And the overall achieved confidence theoretically would be 95 that we aim to achieve. So let's have a look further. I don't want to reallocate the samples at this moment. And we can finish our survey. The pest, let's say, was not found. So we are in the last conclusion page of the repest tool. So here we can also insert the actual numbers of our survey. So let's say these theoretical sample sizes were a bit too many for our resources, and actually we could take 120 samples from the high risk area, and let's say 200 samples from the baseline area. And now we can calculate the confidence that we achieve in reality. So theoretical sample size given in here, the actual sample size given in here, which means that if in reality we have taken less samples that we designed, that means also the confidence level will be lower that we aim to achieve. Also in here, you can download a report of the survey design in two different formats. You have the Excel file in Eurofit format and another format with all the inserted data in the application. But I wanted to show another feature that's called the convenience sampling. If we go back to the estimation of sample size, so by default, if we are not using the convenience sampling, so by default the confidence level will be the same for both of the risk levels that's calculated. But we can also say that we want to use this convenience sampling, and for example, we want to take this, the same amount of samples from both of these risk levels. And we can also do that, and that now we get that we should take 190 samples from both of these risk levels. Of course, the confidence now differs among the risk levels, but the design prevalence and the confidence level will be the same. And the overall sample size will also have decreased a bit because now we're focusing more on high risk levels, and we can even put more focus on the high risk level, saying, for example, that we want to take twice as many samples from the high risk level than from the baseline. Let's see what we get. Now you see that from the high risk level, we should take 224 samples compared to baseline with 112 samples. And again, the difference between the confidence level of these risk levels is now much bigger. So we can even go further and say we want to take 5 times as many samples from the high risk area than from the baseline. We could even try with 10 times. But you see that the confidence level that we receive from the baseline area now is quite low. And we could even decide to focus only on the high risk area compared to the baseline. And in this case, we end up with 271 samples from the high risk level compared to no samples at all from the baseline. So returning to the presentation, here's an overview table of the results of the sample size calculations with repest, and you can see really that there is more and more reduction in total sample size in the more we focus on the high risk areas, and allocating more samples to the high risk areas using convenient sampling will reduce the overall sample size, while still maintaining the same reliability of the survey design.
But Mart, when you are focusing only on the high-risk areas, then the majority of the survey area will not be covered by the survey at all, right?
You're absolutely right, and that's why you should be very careful when excluding the baseline area. But I think that for this pest, for Rhagoletis pomonella, it could be justified because the pest is not known to be present in the EU, so that means we could focus more on the entry sites, and the sampling method is rather ineffective trapping. That means the sample size is higher than compared to the other priority fruit flies. And this would be one way to use the available resources the most effectively. And also one possibility could be to include the baseline survey area in the survey design every second year and not every year. So you could have this kind of different survey designs according to the year. So that would be one possibility. And last but not least, on this last slide, I wanted to show that also in ISPM 26, which is a standard for the establishment of pest-free areas for fruit flies, there is also a trap density suggested for Rhagoletis SPP, and that would be suitable for the establishment of pest-free areas. The standard suggests that trap density of 1 trap per 1 square kilometer would be sufficient for production areas, and 4 to 12 traps would be sufficient for points of entry or these high-risk areas. So using these suggested trap densities within our example, we would result in between 206 to 222 samples. But it's important to keep in mind that this kind of lower sample size, according to the ISPM 26, would be sufficient for declaration of pest freedom in the survey area, but might not be extensive enough for early detection of the pests, so that in case of a finding, the infestation could still be eradicated. So that's really why it's important to understand the difference and to understand the different parameters and really think about what these values actually mean. But I think that was the last slide of our presentation. So thank you very much.
Thank you very much. Thank you very much, Josep. Thank you very much, Mart. I'd like to say sorry again for all the technical and connection issues we had today. We were sorry. We'll try to solve it for the next time. Before we finish, we would like to share with you the second quiz of today with the Mentimeter. It's not an exam. It's just a small test to see if you understood the presentation. So now you should start seeing in your screen this second poll presentation. As I said before, you can either scan the QR code that you have in the screen or go to www.menti.com and introduce the code that you're seeing in the upper side of the slide. Is 63282098. So let's see if we go for the first question of today's poll. Okay, so first question. Contrary to other fruit flies listed as priority pests for the EU Rhagoletis pomonella, so you have 4 options. I see some of you are answering already. For those that couldn't go into the Mentimeter, you can put www.menti.com and introduce the code that you see in the upper side of the slide, 63282098. Okay, I give you a couple seconds to finish answering this first question. Okay, so we have the results. Most of you chose the D. All of the above answers are false. We'll see the experts' correct answer at the end of this test. So let's move on to the second question. Detection surveys for Rhagoletis pomonella in the EU should focus on apples. Option B should focus on adults and therefore could be based on either fruit sampling or female trapping. Option C should emphasize the sampling of areas beyond 600 meters from possible entry points. Or option D, all of the above answers are true. Okay, I see you're answering already. I give you a couple seconds to finish answering this second question. As I said, after the questions, the experts will give you the correct answer. So I see most of you answer again the option D. All of the above answers are true. Let's move to the third question. What is the inspection unit when trapping is used as detection method in the field? Option A, a single tree. Option B, a group of trees. Option C, area covered by one trap. Or option D, the pest itself. Okay, so most of you answer option C, area covered by one trap. Let's move to the last question. To use a risk factor, we need to know, option A, the different levels associated with the risk factor. Option B, the proportion of the population the risk level applies. Option C, the relative risk of each risk level. Or option D, all of the above answers are true. Okay. So I give you a couple seconds to finish. And most of you answer again the option D. All of the above answers are true. So let's see what Giuseppe and Mart think about the answers. And here you can see the results. So if you want to explain, Mart and Giuseppe.
Okay, I can start with the first two questions. And the right answer to question 1 was option B. Because contrary to other fruit flies listed as priority pests for the EU, Rhagoletis pomonella is univoltine and has a winter diapause, which preclude the application of the SIT as well as of MIT. So that's why it was B. I think that was not the most chosen answer. And in the case of the second question, the right answer was A. So detection surveys should focus on apples. Apples are the recommended host for detection according to EFSA. Although detection should focus on adults, fruit sampling doesn't allow the unambiguous identification of the infesting larvae, and they are rearing to adulthood because of the univoltinism and the diapause of pomonella, and therefore cannot be used for this purpose. Additionally, because pomonella is a lazy species, which spreads at a maximum of 600 meters annually, sampling efforts should concentrate within that radius from possible entry points of facilities where apples imported from North America are processed, not beyond that. That was the baseline that, as Mart said, could be sampled maybe only once every 2 years.
As for the question 3, what is the inspection unit when trapping is used as detection method in the field? The correct answer is area covered by one trap, and I think most of the participants were correct with that answer. So it's really the area covered by one trap depending on the attractive range of the lures of the trap itself. And question 4, to use a risk factor, all of the above answers were true. That were the first 3 options, and I think, well, in a way, all of the answers were true, but option D was the most true. And I think most of the participants again answered correctly. You need both of these 3 elements that were described within the 3 options.
Okay, so thank you again. We are getting to the end of the webinar. I want to thank you all for attending. And again, sorry for all the connection and the technical issues we have today. In case we did not manage to answer all your questions, you will find them published on the EU Academy. Please note that similar questions will be grouped in where possible. The recordings of today's event will be able on EFSA's official YouTube channel and in the EU Academy with subtitles in 4 different languages: English, Spanish, French, and Arabic. Next webinars, as you see in your screen, will take place after the summer break. It will be group 2. The first one is September 18th, Anaplophora kinesis. Then in September 30th, we will have Anaplophora glabripenis. And in October 27th, we will have Aromia bungi. Please take a few minutes to scan the QR code you're seeing in your screen right now. It's a evaluation survey. If you are unable to scan the QR code now, after the event, you will receive a link to access the Microsoft survey form. Your feedback is essential to improve our future events. Sorry for today's webinar. We will try this not to happen again. And well, I want to thank you everyone for attending this webinar, and thank you once again, Mart and Giuseppe, for the presentations. We have now reached the end of the session, and I hope you all have learned about this pest and are now well informed about it. This is the last webinar before the summer break, even though our computers, I think they went to summer break before us. As I mentioned, in the previous slide, the next webinar is scheduled for September 18th. I hope you all can attend, and thank you very much and wish you all a great summer.